this is about hybridoma technology and monoclonal antibody. Antibody, right now you may be knowing what is an antibody. So in the case of monoclonal antibody, these are antibodies that are identical. That is identical means same, similar. They have similar property, produced by a single clone of immune cell. What is a clone? Then a group of cells produced from same gene, we can call as a clone. They all have a same type of gene. So that's because they have a same type of gene, they will be appearing very similar, or they will be having similar genetic character. So monoclonal antibodies are antibodies that are produced from a single clone of cell. That is, one cell will multiply into two will be multiplying into 4. I will show you how a clone of cell will be appearing. Here you can find an image of a clone of C. This is how a clone of cells will be usually finding. One cell will be dividing into 2. From that cell we will be having large number of new cells. All will be having the same feature. So what is the genetic composition of this cell will be exhibited by all these cells. Such a group of cells are known as a clone of cell. So in the case of a monoclonal antibody, all these all these antibodies are produced from a clone or from a similar group of cell. That is the main feature of a monoclonal antibody. Nowadays they are very important in many areas of medicine, like diagnostic medicine, therapeutic area of the medicine, that is treatment area of the medicine, and also in the research area of the medicine. We are utilizing monoclonal antibodies a lot. So coming to the main features of monoclonal antibody, here you can find that there are 3 important properties for a monoclonal antibody. There is, first one is they are highly specific to a particular antigen, which makes them valuable for targeting specific cell or protein cell. For example, you have one person has a lung cancer, and we are using a monoclonal antibody against cancerous cells affecting lungs. Then that person, we can inject that monoclonal antibody into him or ask him to take a tablet or medicine. And what this monoclonal antibody will be doing is that they will go to the lungs and kill all the cancerous cells. Right now what is happening is that we are using a crude approach. For example, when you are using a chemotherapy, it will be killing both cancerous cell as well as normal cell. So that is the approach of that is the common approach. But in the case of a monoclonal antibody, they are highly specific. So whatever cell which we have targeted, only that particular type of cell will be affected. Second property is that homogeneity. They are derived from a single clone, and all the monoclonal antibody are identical in structure and function. So they are highly similar, they have same structure and same function. So that is another peculiarity, that is homogeneity. That property of this having same structure and function is known as homogeneity. Third feature is that they can production. Monoclonal antibodies can be produced in large quantities and can be engineered to enhance their property. By genetic engineering, we can produce large quantities of monoclonal antibody, and so that is one important feature which makes them very valuable in medicine. So we have the ability by genetic engineering to produce large quantities of monoclonal antibody, improving their binding affinity or reducing their, that is, by utilizing our research method, we can improve the effectiveness of the antibody also. So these are some important features of monoclonal antibody. So monoclonal antibodies are always closely associated with the hybridoma technology. So here there are 3 important features. We have to understand what is a monoclonal antibody. and what are the three important characteristics of monoclonal antibody. Then here we have another technology. This, along with this technology, we usually associate monoclonal antibody, that is hybridoma technology. It is a process used to produce monoclonal antibody. So if you get a question, what is hybridoma? It is a technology which we use to produce monoclonal antibody. It involves fusion of a specific type of immune cell or a B cell with a myeloma cell or a cancerous cell. That is, two types of cell. One is a normal immune cell or a B cell will be combined with a myeloma or a cancerous cell, creating a hybrid cell line that can proliferate indefinitely and produce a large amount of specific antibody. That is, in the case of this normal cell is capable of producing antibody. One important feature of the myeloma cell or a cancerous cell is that they can multiply unlimitedly. So normally most cell will be stop multiplying after a certain number of cell division. But in the case of a cancerous cell, this stopping of cell division is not taking place. That is, a cancerous cell will keep on multiplying or keep on undergoing cell division indefinitely. So there is no stop. That is why when one part of the body is having a cancer, we can see that that part is bulged or enlarged because of the continuous multiplication or continuous cell division happening in a cancerous cell. So this is how the this quality of the cancerous cell is also employed in hybridoma technology. I will show you some pictures of how hybridoma technology is utilized. So here you can see that the hybridoma technology, see, this is how the hybridoma technology is normally they are utilizing. The picture is very short. Let me take another picture. See, this picture is somewhat better. So here there are two types of cell. So one is a normal plasma cell. Plasma cell means any cell that is producing antibody. Other is a myeloma cell. Under the myeloma cell they have written immortal. That is, they have the property of keep multiplying without any end. But whereas in the case of a normal cell, they will stop multiplying or stop cell division after some 50 times or 100 times. But that is not happening in the case of a cancerous cell. So what we are doing is that we are actually combining the properties of a myeloma cell and a cancerous cell in a single cell. So what we do is that we take this cell and we will be combining it and forming this plasma cell and myeloma cell are combined, and as a result we will be getting a hybridoma, a hybrid cell. That hybrid cell will be then then cultured in a medium, and after culturing we will be getting a lot of we are getting a lot of this hybridoma cell. That hybridoma cell will be able to produce large number of antibodies, and that antibodies will be all having same structure and same function. So that is known as a monoclonal antibody. So here you can see that in the picture it is very clearly shown that hybridoma technology is a technology used to develop a large number of monoclonal antibody very easily. So it is this technology that we are employing in the case of the monoclonal antibody. So what are the steps in a hybridoma technology? One is immunization, cell fusion, selection, then screening and cloning, etc. These are some of the important steps. Coming to the immunization, an animal, typically a mouse, is immunized with an antigen. So if you want to develop a monoclonal antibody from one animal, normally what we are doing is that we take the virus, inject that virus in. For example, you want to develop a monoclonal antibody against coronavirus. What we will do, we will take the coronavirus and introduce this coronavirus into a rat. So after some time you can see that rat will be producing a lot of B cells which are capable of producing antibody against coronavirus. We collect some of those kind of lymphocyte or those type of B cell. The next step is that cell fusion. After sufficient immune response is achieved, B cell from that rat will be collected from the spleen of the animal. by killing it. Then these B cells are then fused with the myeloma cell using a fusogenic agent such as polyethylene glycol. Here you might have seen that they have shown some material, polyethylene glycol. So using polyethylene glycol, here they have also mentioned. Polyethylene glycol is a chemical that will be facilitating the combination or union of a B cell and a myeloma cell. These B cells are then fused with the myeloma cell using a fusogenic meaning something that facilitates fusion or union, and that is known as a polyethylene glycol. And as a result, we will be getting a combined cell called as hybrid cell, otherwise known as hybridoma. Now, third step is that the hybridoma cells are cultured in a selective medium, and that medium is known as the hat medium. Here also you can see that this is the hat medium. So hybridoma cells will be then cultured in a hat medium, and what is happening is that allows only hybridoma to survive while eliminating unfused myeloma cell and non-fused B cell. So in the case of a hat medium, their ability is that they will only promote the development or culture of hybridoma cell, whereas it will not facilitate the development of normal non-hybridoma cell. The resulting hybridoma are a clone that produce the desired antibody. So the hybridoma cell will be having two features. One, to produce antibody. Second feature is that they will be able to multiply indefinitely. So that is the third step in hybridoma technology. Then fourth step is screening. The supernatant hybridoma culture is screened to identify those producing antibody that can bind to the antibody. So here in this screening, we are making sure that this particular hybridoma cell are effective against a particular organism or a particular microorganism, in that they will be producing large number of similar type of antibody. This can be done using various assays such as ELISA, etc. Then cloning. So in this cloning, the positive hybridoma are further cloned to ensure that they are monoclonal, meaning all cells in the cells originate from a single hybridoma. That is, all the cells which are producing, here you can see that all these cells are actually developing from one particular hybridoma cell. So they will be having this. Here you can see that different types of cells are, but we are taking only one cell. That one cell is multiplied several times, and from that we are getting a lot of monoclonal antibody. So this is how we are actually developing lot of monoclonal antibody. Then expansion and characterization. The selected hybridomes are expanded to produce large quantities of desired monoclonal antibody. They are then characterized to determine whether they are specific to that particular antigen. They have affinity or attraction. They have different types of isotopes are also there. Such possibilities are also studied. Then what are the applications of monoclonal antibody? In three areas we are using in therapeutic. Therapeutic means treatment. Second is diagnostic, then research also. Monoclonal antibodies are used to treat variety of diseases such as cancer, autoimmune disease and infectious disease. So that is one important advantage of that. They can mark by neutralizing pathogen and marking them for destruction by immune system or by blocking cell signaling pathway. That is another important advantage of monoclonal antibody. Second property or second application of monoclonal antibody is in the diagnostic. So coming to the diagnostic, monoclonal antibodies are widely used in diagnostic tests, including those for dictating disease like HIV, cancer markers and various infections. So that is for dictating HIV we can use a monoclonal antibody. For dictating cancer we can use monoclonal antibody. They can be used in imaging techniques like PET scan when conjugated or combined with a radioactive isotope. So in the case of a PET scan, what we do is that we will be combining this monoclonal antibody with a radioactive isotope, so that it will be able to reach that particular cell and also show what are the different types of blocks observing in that. The research monoclonal antibodies are essential tool in research for protein detection, quantification and purification. So these three are the important properties, important applications of monoclonal antibody in research. They are used in Western blotting, immunohistochemistry and flow cytometry. So these are some of the important applications of monoclonal antibody. We can see that they are used in treatment, they are used in diagnosis, and they are also used in advanced research. Then coming to the last one, the recent advancement in monoclonal antibody include humanization. Humanization means to reduce immunogenicity of marine monoclonal antibody. Techniques like CDR grafting have been developed to create humanized or that is normally we will be developing monoclonal antibody from some animal. When this animal monoclonal antibodies are injected into the human being, that will sometimes create allergic reaction. So to avoid that, we are now trying to make human-like monoclonal antibody, humanized monoclonal antibody. Then bispecific antibody. These are engineered to bind with the two different type of antigen, providing unique therapeutic advantage. That is, in the case of treatment, there are a lot of advantages if two different type of antigens can be combined. Then antibody drug conjugate. It can be conjugated with a cytotoxic drug, allowing for the targeted delivery. That is, for killing a particular type of cancerous cell, we can utilize this monoclonal antibody. Then there are a lot of complications still, lot of difficulties still. One is cost of production. Producing high quality monoclonal antibody can be expensive and also time consuming. That is one problem. Regulatory hurdle. Ensuring the safety and efficacy of rigorous testing is essential. That can prolong developmental timelines. So that is very important, because most of the time this kind of vaccination or or virus related things are done very slowly. So that will be the problem. Then immunogenicity. While humanized monoclonal antibody are less immunogenic, there remains a serious risk of immune response in some patients. In some patients, even taking a vaccine can result in death. So this is an idea regarding the different type of monoclonal antibodies also.